Today's discussion will be all about the abnormalities or defects of the sperm of the bull, stallion, and boar. The sperm consists of the head, neck, middle piece, and tail, all of which are covered by a plasma membrane. The head consists of a cap-like structure called the acrosome, which contains digestive enzymes that help in the fertilization of the ovum. It also contains a condensed haploid nucleus. The middle piece consists of many mitochondria, which provide energy to the sperm for locomotion. It also contains the centrioles, which aid in flagellum formation and link the head and tail to control its beating. The sperm also consists of the tail, which functions for the motility of the sperm. Sperm numbers and quality can be compromised by a variety of factors which include genetic, traumatic, nutritional, toxic, or infectious considerations. Apart from genetic sperm defects and specific spermatoxic agents, both of which are relatively rare, the most common factor is spermatogenic stress. It is established that any environmental stress sufficient to cause elevation in circulating cortisol is sufficient to affect sperm morphology. A cascade of linked developmental processes occurs during spermiogenesis that are testosterone-dependent, such as formation of the acrosome from the Golgi apparatus, compaction of sperm chromatin, mitochondrial and centriole organization to form the flagellum, and initiation of cytoplasm resorption. In a study, a single acidotic event was followed sequentially by elevated cortisol, reduced FSH and testosterone consequent with increased sperm abnormalities in the subsequent weeks. Similar sequential appearance of spermatozoal abnormalities were observed following transport and relocation, or dexamethasone. Analogous abnormalities have also been observed following exposure to heat, whether due to obesity, scrotal abnormality, climate, or fever. Sperm abnormalities can be classified according to the location of the defect. It is further classified into two: primary sperm abnormalities and the secondary sperm abnormalities. Primary sperm abnormalities include defects that are located in the head of the sperm. Primary abnormalities are caused by a failure during the formation of the cell in the testes. Secondary sperm abnormalities include defects associated with the neck, midpiece, and tail. The presence of cytoplasmic droplets is also included. Secondary abnormalities occur as the sperm travel through the epididymis. Now, let's move on to the morphology of bull spermatozoa. Figure 1 shows the normal morphology of the bull spermatozoa. A normal, healthy sperm has the following characteristics: an oval-shaped head that is symmetrical and smooth, an intact midpiece and straight neck, a long, straight, uncoiled, and undamaged tail. The standard threshold of a normal sperm should be greater than 70%. This is the minimum threshold for a bull to be classified as a satisfactory potential breeder. Less than the given value denotes failure in morphology. Now, we are moving on to the primary abnormalities of the bull spermatozoa. Number 1 shows a decapitated sperm head. This is a tailless or a caudate sperm which resulted from acrosomal defect. It is typically seen in necrosis and may be genetic. Separation of the sperm head and tail can be caused by a number of adverse factors affecting either spermiogenesis or sperm maturation. However, a specific sterilizing form has been reported in several cattle breeds: Guernsey, Hereford, Swedish, Red and White, where it has been associated with sterility. It can be differentiated from detached loose head by the large number of vigorously moving tails in the fresh specimen and the presence of the proximal droplet still attached to the tail. This trait, when it occurs, affects 80 to 100% of sperm in the ejaculate. This also arises with testicular degeneration, or hypoplasia, inflamed ampullae, or epididymis, heat stress, and more usually, as a sign of a rusty load. Number 2 shows a macrocephalic sperm head. Macrocephaly refers to when the sperm has an enlarged, irregularly shaped head. This usually indicates deficiency in chromatin condensation. As such, this sperm often carries extra chromosomes in its head. This sperm can exist in two types, with multiple tails, or aflagellate, or no tail at all. Macrocephalic sperm may be caused by homozygous mutation of the aurora kinase C gene. This means that because this kind of sperm abnormality may be genetic, it can be passed on to the offspring. Moving on to number 3. The picture shows a microcephalic sperm with stump tail. This is a condition wherein the sperm head is smaller than normal. Microcephalic spermatozoa have small and irregularly shaped acrosomes that usually are not in contact with the nucleus. Acrosome abnormalities are common in these sperm, and as it is small, there is minimal to no paternal DNA content. Next, we have the pyriform sperm head. The head becomes narrow in the postacrosomal region. Young bulls up to 2 years old and in good condition display a greater likelihood of recovery from this condition than do older bulls. This condition is particularly seen in young over-fat bulls. Fertility was related to the severity of pyriformity of the head. A moderate degree of pyriformity, in the absence of other signs of disturbed spermatogenesis, is not detrimental to fertility. However, extreme tapering in the postacrosomal region results in significant reductions in fertility. Pyriformity is considered only partially compensable. Pyriform heads are induced in bulls following stress, such as dexamethasone treatment and scrotal insulation. The fifth primary abnormality is a round-headed sperm as opposed to sperm heads that are usually of oval shape. Globozoospermia, or round-headed sperm condition, is an abnormal sperm morphology that indicates either there is an absence of the acrosome or the sperm is missing inner parts of its head responsible for activating or turning on the egg and starting the fertilization process. Sperm can break down prematurely, decondensing head sperm. The next one is the round head with double tail. It is another type of globozoospermia in which the sperm contains 2 tails instead of the normally single elongated one. This is a rare type of abnormality. Next slide contains the ruffled acrosome. This is a sperm with irregular staining acrosome leading to a wrinkled or ruffled appearance. This condition has been reported in subfertile bulls where they were linked with the knobbed acrosome defect. A genetic basis was suggested by the occurrence of the 3 defects in 4 sons of a subfertile Holstein sire in one study. Tapered sperm head. Tapered head sperm are sperm with cigar-shaped heads, characterized by its elongated with a narrow base structure. This condition indicates the presence of varicocele. This can be associated with constant exposure of the scrotum to high-temperature locations. These tapered head sperm often contain abnormal chromatin or packaging of the paternal DNA genetic material. Abnormal number of sperm chromosomes called aneuploidy has been shown in tapered head sperm. Craters or diadem defect are small red-colored spots located along the nuclear ring of the sperm head, just like a row of pearls or necklace or diadem. The appearance of diadem defect in sperm heads only serves as an early sign of various forms of head abnormalities occurring as a cascade of sperm defects which eventually progresses to vacuoles craters. These sperm head abnormalities are mainly due to problems in DNA chromatin condensation and organization during spermiogenesis, or the sperm differentiation phase, when changes to chemical chromatin bonds are particularly vulnerable to oxidative damage. This condition is associated with testicular overheating and stress, and it causes lowered motility. The double-headed sperm are sperm with 2 heads, also called as duplicate sperm. This sperm is incapable of penetrating an egg due to low motility. This condition is often linked to exposure to toxic chemicals, heavy metals like cesium, smoke, or high prolactin hormone in the male animals. Next, we are moving on to the secondary abnormalities found in the sperm of bulls. First, there is the proximal cytoplasmic, protoplasmic droplet. These are droplets located near the neck or the midpiece of the sperm tail. It represents excess cytoplasm that has not been eliminated. These are normally observed in the pubertal bull, with incidence decreasing with age. In the mature bull, they indicate abnormal spermiogenesis or epididymal function. Next, there is the distal cytoplasmic droplet. These are droplets located at the distal end of the midpiece. Sperm with distal droplets will lose the droplet if left in a water bath for 15 to 30 minutes, or if gently agitated. There is reduced motility and fertility potential. The presence of a cytoplasmic droplet, whether in the proximal or distal position, may be an indication that the sperm has not acquired essential binding proteins from the seminal vesicle fluid. These binding proteins are essential for the sperm to bind to the zona pellucida. For this reason, it is important that massage of the ampullae and seminal vesicles is sufficient to elicit a quantity of seminal fluid during the collection process. Moving on to translocating cytoplasmic droplets. These are droplets that are actively moving along the sperm tail from a more proximal location to a more distal position as the sperm matures. This may have reduced fertility. Tail opening following droplet translocation. Following the translocation of the droplets, a tail opening may be observed, which is essentially an area where the cytoplasmic material has been reduced or removed, creating a clearer space along the tail. This indicates sperm maturity and efficient motility. Coiled tail with droplet. It is also known as a coiled flagellum, and it occurs due to damages in tail. In this condition, tail may be twisted or spiraled instead of being straight and elongated, which results to the sperm unable to swim well. This also indicates the presence of bacteria. Coiled double tail. It is a sperm that has two tails in a spiral or twisted shape. Due to this abnormality, sperm motility is reduced. This can be caused by genetic mutations. Next is the DAG defect. It is a strong folding, coiling, and fracture of the distal part of the sperm midpiece. It is named after the Jersey bull in which the defect was first identified. The defect may reflect disturbance in the testis or epididymis and suggested a genetic cause. This can be an inherited defect due to autosomal recessive factor that is associated with impaired fertility in bulls. Dog defect was first identified in Danish Jersey. Bulls later were reported in Hereford bulls. The next sperm defect is folded tail. It is an abnormality that shows a strong folding of the tail, which affects sperm motility. This could be due to genetic defect or environmental stressors. The sperm tail can also be filamentous, where it has an abnormally thin, elongated, or thread-like tail. It results from genetic mutations or chromosomal abnormalities, although temperature or varicocele also contribute. The second photo shows a sperm that has two tails connected in a single head. It can either be hereditary or occur as random mutations during spermatogenesis. Corkscrew midpiece with droplet. It is a condition wherein there is irregular distribution of lumps of mitochondrial sheath, resembling a corkscrew as compared to normal cylindrical appearance. This occurs together with a high incidence of proximal droplets. This is commonly seen in aged bulls in several exotic breeds. Similar defects have been reported in bulls following bovine ephemeral fever and in bulls undergoing gossypole spermatoxicity. This could also be due to progressive testicular degeneration resulting in decline in sperm count. Recovery is rare. Next is bent tail. As you can see in the picture, the tail of the sperm is bent, forming a perpendicular angle from the shaft as compared to its normal straight structure. This could be due to normal tail movement or to either a structural defect or physical disruption. Now that we've covered the morphology and abnormalities of bull spermatozoa, we'll shift our focus to equine spermatozoa. While there are similarities between species, equine sperm have their own specific characteristics and unique challenges in terms of morphology and fertility. Let's begin by examining what constitutes normal sperm morphology in horses, and then move on to common abnormalities. A normal, healthy equine sperm cell is essential for successful fertilization. It features a symmetrical, oval-shaped head that is smooth, with an intact midpiece and a straight neck. The tail should be long, straight, uncoiled, and undamaged. In terms of quality assessment, the standard threshold for normal sperm morphology should exceed 70%. If the percentage of normal sperm falls below this value, it suggests abnormalities in morphology that may reduce fertility. When considering head abnormalities in spermatozoa, there are several types to be aware of. A loose head is one that has detached from the midpiece and tail. Without these essential connecting structures, the sperm becomes non-functional and cannot contribute to fertilization. Small and narrow heads are characterized by reduced size and an elongated shape. These abnormalities often result from genetic defects, nutritional deficiencies, or environmental stress. Another abnormality to watch for is an elongated head. In this case, the head is longer and more tapered than normal, which may stem from genetic factors or abnormal spermatogenesis. Pieriform, or pear-shaped heads, are also abnormal and can result from genetic anomalies or defects in spermiogenesis. Macrocephalic sperm, or sperm with enlarged heads, have an abnormally large, rounded appearance. These are often linked to genetic mutations or chromosomal abnormalities. On the other end of the spectrum, microcephalic sperm have significantly smaller heads, which is usually due to genetic defects or impaired spermatogenesis. A rarer defect is the presence of an additional acrosomal cap, referred to as an additional cap. This anomaly involves the formation of a double acrosomal cap structure on the sperm head and is caused by defects in acrosomal formation during spermatogenesis. Secondary abnormalities of the sperm neck can indicate issues during spermiogenesis. One common abnormality is the retention of cytoplasmic droplets. These droplets can appear near the neck, referred to as proximal cytoplasmic droplets, or further down near the midpiece, called distal droplets. Both types suggest incomplete maturation of the sperm and can be linked to stress or abnormal spermiogenesis. Another neck abnormality is swelling. In some cases, small red spots form along the nuclear ring, resembling a necklace or diadem. This diadem defect is associated with testicular overheating and stress, which reduces sperm motility. In other cases, sperm may have split or divided necks, with two heads attached to a single neck. This defect significantly impacts the sperm's motility and is often caused by exposure to toxic chemicals or heavy metals. An irregularly shaped neck is another common secondary abnormality. The neck may be misshapen or asymmetrical, often due to genetic defects or disruptions in spermatogenesis. Another issue is a broken neck, where the neck appears angled or fractured. This can result from mechanical trauma during sperm collection or processing, or from abnormal development. Thread-like abnormalities are characterized by small red spots along the nuclear ring, resembling a row of pearls or a diadem. Like other abnormalities associated with the diadem defect, this issue is linked to testicular overheating and stress, which reduces motility and affects fertility. Tail abnormalities also play a significant role in sperm health. A two-headed sperm is a rare defect in which two heads are connected to a single tail. This typically results from genetic issues during spermatogenesis. Another abnormality is a thread-like tail, which is abnormally thin and elongated due to issues in tail development during spermatogenesis. A sperm with a single loop in its tail exhibits a noticeable curve or loop that deviates from the normal straight tail structure. This defect arises from abnormalities in tail development. Another tail abnormality is DAG, or double axoneme hegerminal tail, which results in a thickened appearance due to the presence of two axonemes. These issues stem from defects in spermatogenesis. A coiled tail is a severe abnormality in which the tail forms a spiral or corkscrew shape. This defect impairs motility and is often caused by developmental issues or environmental stressors. A similar defect occurs when the tail coils around the sperm head, which further reduces the sperm's ability to fertilize an egg. Lastly, a bent tail is another common abnormality. This defect is marked by a noticeable curve or angle along the tail's length. The cause can be either structural defects during sperm development or mechanical trauma that occurs during collection or processing. Now we are moving on to the morphology of the boar spermatozoa. The picture shows the normal morphology of a boar's sperm. The spermatozoon of the boar closely resembles that of other domestic species. A boar spermatozoon considered normal must have three perfectly defined structures: oval head, without vacuoles or irregularities in outline, a midpiece or neck of adequate thickness and length, and a thin, long, and mobile tail. As usual, the standard threshold of the sperm's morphology should be greater than 70%. The following slides show morphologic characteristics similar as that of the bull and stallion spermatozoon. A spermatozoon is considered to be morphologically abnormal when it presents one or more of these altered structures. This includes a microcephalic head, a giant or macrocephalic head, and an elongated head. Boar spermatozoon can also oftentimes exhibit short or even detached heads. All the aforementioned defects indicate acrosomal abnormalities and a negative effect in the genetic material of the sperm. Boars can also have knobbed acrosomes. The major manifestations of this abnormality are 1. A refractile, thickened acrosomal apex, and 2. An indented sperm apex. In boars, the defect has been associated with gene aberrations on chromosome and with both dominant and sex-linked recessive modes of transmission. Affected boars produce ejaculates that contain spermatozoa with knobbed acrosomes, resulting in low fertility. This abnormality may be heritable or arise following a stress event. There are also pyriform heads, where there is narrowing at the post-acrosomal region of the sperm, resembling that of a pear, as well as other types of primary abnormalities that could be rare. Aside from the primary abnormalities, there are also secondary abnormalities exhibited by the boar spermatozoon, which could either affect the midpiece or the tail. This slide shows a broken tail, where the tail is snapped off from the head. The other photo shows a sperm with an abaxial tail. Abaxial tails are observed when the insertion of the tail to the head is considered to be off-center. They are relatively common in some species of boars. We also have the bent tail, translocating droplet, as well as the coiled tail. As mentioned before, these abnormalities could either be due to genetic defects or environmental influences, with stress also being a predisposing factor. Boars can also have spermatozoa with double midpieces, along with other rare midpiece abnormalities which are shown on the second picture of the slide. The most common abnormalities are the presence of proximal and distal cytoplasmic droplets in the boar's sperm. In both cases, these are secondary malformations that occur during sperm maturation in the epididymis. Immature spermatozoa with proximal cytoplasmic droplets originate in the testicles, and during their journey to the epididymis, the cytoplasmic droplet moves towards Jensen's ring. Once sperm with distal cytoplasmic droplets reach the epididymis tail, they lose the cytoplasmic droplet and take on the appearance of a mature sperm. Thus, the greater or lesser presence in the ejaculate of immature spermatozoa allows us to ascertain the extent to which the sperm epididymal maturation has progressed. Therefore, distal drops provide a better prognosis for the viability and fertilizing capacity of sperm than the proximal type, because they are cells which have developed further in their maturation process. The presence of immature spermatozoa, which show protoplasmic droplets on their tails, indicates that a boar has been overworked. The origin of abnormal forms, especially the secondary type and the lack of maturation of the spermatozoa, may be due to a reduction in flow of testosterone at the testicular level. This is often due to stressful situations. They may also have an anatomical origin, such as the presence of a varicocele. In this slide, we will review examples of combined abnormalities seen in boar spermatozoa. Picture 1 shows a sperm with a nuclear vacuole and a distal droplet, both of which indicate incomplete maturation and possible fertility issues. Picture 2 highlights more complex abnormalities, including a small head, pyriform shape, and both abaxial and bent tail defects. Lastly, Picture 3 presents a range of abnormalities: one sperm with a pyriform head and an abaxial tail, another with a problematic vacuole, and one with a pyriform head and distal droplet, all showing multiple structural issues that could impair fertility. Continuing with more examples, Picture 1 shows a sperm with an elongated head and a proximal droplet, suggesting poor maturation. The second picture illustrates a knobbed acrosome, a condition where the acrosome appears irregular or misshapen, potentially interfering with the sperm's ability to penetrate an egg. In this slide, we observe a range of abnormalities. Picture 1 presents sperm with abaxial tails and an abnormal midpiece, both of which could affect motility. In Picture 2, we see several types of defects, including a broken tail, elongated tail, pyriform head, and a sperm with an abnormal midpiece. Additionally, there's a sperm with a large head and distal droplet, highlighting the variety of structural abnormalities that can occur. Finally, in this slide, we see additional examples of boar sperm abnormalities. Picture 1 shows a sperm with a long head and coiled tail, both of which could significantly reduce motility. Picture 2 highlights a sperm with an abnormal midpiece, and Picture 3 shows a sperm with both a long head and abaxial insertion, further illustrating how combined defects can affect the overall function of the spermatozoon.